that these presentations represent, as well as the way they showcase the variety of projects that can be undertaken during an undergraduate career at Augustana. From a paper originally written in an FYS class and developed during a study abroad, to collaboration with faculty members on laboratory experiments and archival research. After each presentation, there will be 5 minutes for questions. You can email your questions at any time during the presentation to symposium@oli.augie.edu, and they will be read by one of the moderators. We won't attach your name to them, so they'll be read anonymously. Thank you.
All right. Hello, everyone. My name is Ritterbusch. I'm a senior biochemistry and sociology double major, and I'll be going to medical school next year. And so today I'm going to be presenting a project that I worked on the last couple of years, 2 years, in medical sociology. And so this work was done kind of as a team with Dr. Ly-Donovan as our faculty mentor, and then 2 other students who have graduated, Dan Schmidtman and Evan Meyer. And so they graduated, and they're also going to medical school next year. And so the project is titled The Organizational Priorities of the South Dakota State Medical Association from 1882 to 1956. Just a little outline of how this is going to go. So I'll talk about how I got involved in the project before getting into the research findings like methodology, and then just a little bit about some of the benefits that I found doing research here at Augustana. So I came in as a freshman as a biochemistry major. I knew I wanted to go to medical school, but at the time I wasn't really interested in sociology or social science research. I kind of knew that research was important for going to medical school, and so the summer after my sophomore year, I did a project in physical chemistry. So a picture up here from that is obviously very different. And kind of after I did that, I figured that that was my one research experience. I didn't really think I'd be getting into anything else. Separately, I took Sociology 110, and that's when I started to get interested in sociology, and I had a sociology major. And I really started to become interested in some of the social influences of health, things like health disparities and health policy, and starting to really consider maybe a career both in like clinical medicine, but also in research, potentially in like public health. So this particular project actually didn't come about from any sort of like formal application process, but rather I just had taken a couple classes with Dr. Ly-Donovan, just through going to office hours, just kind of having informal talks about like my career plans, like what I was interested in. She came to me with this opportunity to join this project, and this was something that had been started just a little bit before by her and those 2 other students, and she asked if I wanted to join, and of course I said yes. I thought it was a really great chance to do something different, something unique, something that a lot of pre-med type students wouldn't really be interested in thinking about. And also just an opportunity to start thinking about medicine outside of like, the typical biomedical, like biochemistry route that I was used to. And so we started in the spring of 2017. So the real aim of this research was that we used archival data and a cultural analysis of that data. We looked at the entire official archive collection from the South Dakota State Medical Association, which is housed here at the Center for Western Studies. And we went in thinking that we would reconstruct key moments from this association's history. So this is kind of what it looked like, a lot of the materials that we were looking at. So things like handwritten notes, meeting minutes, monthly published journals for the association, the journal Lancet, which was something they published yearly. And we really started to look deeper into those. And so at the Center for Western Studies, there's several just boxes, filled with all these materials in different folders. And so to get into the actual research process, the first part was really figuring out what exactly we wanted to look at of all of these materials. There's just so many different pages of things that potentially could be read. And so thinking about the process of it is quite a bit different than what I was used to, probably what most people are accustomed to thinking about when we think of research and more like biology or chemistry. So we started by trying to identify time periods or materials that we thought would be interesting to read closer. So an archived data set has these finding guides that have been created by like the librarians or the historians. And so sort of like a table of contents for a book. These outline sort of what you're going to find in each box and in each folder. And so we use those to really start to identify areas of interest to search deeper. We also looked at things like kind of like overview history books or what was happening in national legislation at the time that might be interesting to compare. At that point, we started to do the content analysis. So we would get together just kind of a couple hours a week whenever we could at the Center for Western Studies and just started reading these. Started identifying which years of interest we wanted to look at, and then when we got together, we would each person would read a different article or letter, and then we kind of used each other to bounce ideas off of and start to think about the data and compare like what we were finding, and in that process we then started to construct a timeline. And so this was just a screenshot of the timeline with we put in significant moments, significant quotes or legislative actions that we saw, and then color coded it so we could look in between each years for themes that were similar to each year. So if they're talking about legislation, if they're talking about Native American health, which is what really the project turned its focus to. So we started kind of drawing some empirical conclusions initially, and that's when we really started to focus in on this surprising lack of mention of Native American healthcare, and we sort of turned our focus to the priorities of the group in context of also how they're talking about Native American healthcare at the time. What we found before jumping into some examples, just as an overview, we saw that the South Dakota State Medical Association was actually very influential in the healthcare policies and regulations in South Dakota. And for them, it really centered around this main theme of establishing legitimacy and institutional power. So today, when you're sick or you're going to get surgery, we really take it for granted the fact that MDs and physicians have a certain amount of status and prestige in our society that there's really just complete trust of going to a physician for medical knowledge. But in the late 1800s and early 1900s, it wasn't necessarily like that, and there's different medical professionals such as chiropractors or osteopaths that people would turn to, or just things like self-medicating for people who wouldn't maybe trust going to a physician. And then in comparison to that, we also looked at how they're thinking about Native Americans, and we found that there was a really surprising and telling lack of consideration of Native Americans by the association during really critical time periods of Native American healthcare policy, particularly at the federal level. And we think this really illustrates how structural inequalities today, the fact that there's just massive disparities in healthcare outcomes for like Native Americans, for example, versus the general population, but these can arise through organizational development. So to get into some examples, one thing we looked at was, again, what they're prioritizing and how they're exactly going about establishing legitimacy and institutional power at this time. One way they framed it is this idea of proper propaganda, as they called it. So we saw a quote from 1924 where So there's only one way to counteract the cults, and that is by education, by means of proper propaganda. So here with cults, they're talking about the chiropractors and osteopaths, and really sort of kind of making the case that, for the MD, for these medical professionals to become sort of the legitimate source of health information in South Dakota, they really had to kind of delineate themselves from those other professions. Again, 6 years later, a quote from a physician, "The osteopaths and the chiropractors and every damn cult want recognition. And here's one on his feet who will not recognize them. I will fight them to the last ditch if it is necessary." So we saw throughout the years that they're talking about this in a similar way and really putting a lot of focus on how to educate, South Dakota, how to really kind of advertise themselves as a legitimate source of medical information. Another thing that we looked at was these sanitation and education efforts. So initially, part of the kind of the cultural analysis of this was to understand what exactly they're talking about when they were frequently mentioning like sanitizing different groups in South Dakota. And really what we found within the context of the time that this was sort of their first public health outreach efforts. This was a time where like the germ theory was just coming around, so people were finally starting to kind of understand how people get sick, the importance of hygiene. And so this quote from them, from minutes from one of their meetings, "The health of the community can be materially influenced by the intelligent teaching and practical demonstration of hygiene and sanitation in the public schools." And so we saw them doing several sorts of outreach projects for a variety of different groups, everything from teachers to women's club to crippled children. And this was really significant when we then compared it to Native Americans because, these groups were marginalized essentially when you consider, for example, like the standing of even the women that far back, or just crippled children. So we did see that there was outreach efforts for certain groups, but we thought, tellingly, there was none of that for Native Americans. And so this was one of the few times we saw mention of Native Americans in discussion. And I think it also kind of exhibits some of the difficulties in just reading and understanding some of the letters and things that they were writing back then. So this was talking about a member who was asking for increased hospital facilities on the reservation for the protection of the Indians and for the removal of contagions from the white settlers. So this is really what we saw as them framing any sort of mention of Native Americans. We definitely didn't see sort of any materialization of this idea of building more facilities on the reservation. Rather, we saw them most of the time framing this as keeping the sickness that was prevalent on the reservations isolated from white settlers. And then another thing that we did was we were able to compare national policy to the priorities of the group at the time to really consider what they were talking about, what they were thinking about, what were they making important versus really significant federal policy changes. And so things like First Indian Hospital on the Sioux Reservation, you might expect that such a prominent group of health professionals would at least mention that in meeting minutes or in notes or journals, but really we saw nothing like that, but rather they would talk, that year they made malpractice insurance a large priority. And we saw that throughout even, the formation of the Indian Health Service to the Transfer Act of 1954, which is another really significant moment in IHS history, where again, just this really telling absence of any mention of Native Americans in the association. And so, to conclude for the research part, we saw that while the SDSMA is a voluntary professional association, they're actually very influential in shaping healthcare policies in South Dakota. They had this increasing amount of power during important changes on the federal level for Native Americans. but they really paid little attention to the needs and policies of Native Americans in South Dakota, despite recognizing that the Native Americans were a large portion of the population and that they were disproportionately sick. And we argue that this is important today when we think about why there are some of the structural issues and organizational issues for healthcare for Native Americans. And then to just change directions, I talked for me about some of the benefits that I found from doing research at Augustana. I mean, first, just being able to do something different with this and learn more historical and sociological methods, things like handling archived data, 100-year-old documents, to using sociological framework to think about healthcare and to think about data. It really also encouraged me to look for more research opportunities, and I was able to last summer go to Massachusetts General Hospital. I did an internship there, and so that's just a picture of me in Boston. And I did a project looking at cerebellum atrophy in multiple sclerosis patients. So I saw that it was obviously a very different type of project than what I was working on here, but definitely sort of a foundation through how to ask good research questions and work with a faculty member and work within a team of researchers. I thought it really set me up well for going on to that opportunity and then to medical school. We also had a couple of really awesome opportunities to travel to present our work and also for publishing. So right now we have a paper under review at Great Plains Research. Last spring actually, we traveled to Yale. So Dr. Ly-Donovan presented our work at Center for Cultural Sociology there. So for myself and other undergraduates, it was really awesome too. We got to visit the medical school, and talk to Yale graduate students. It was really a great experience for us. And then this fall, we traveled to Montreal, Canada, which I also have a picture up there, to the Social Science History Association annual conference. And we really saw that, at both those things, we were the only undergraduate students. And through Augustana, we had a really awesome opportunity to work with a faculty member and to be able to produce work that was really kind of a graduate level and present alongside, PhDs and graduate students. And something for me that came out of not even, a formal application process, but really just kind of showing interest, talking to my, professors, and then taking advantage of the opportunity when it was there. So thank you, and I guess if there's any questions, we'll do that right now.
And it's not too late to send your questions to the email address on the slide. Here's your first question. Are Native Americans still disproportionately underrepresented at the state and national levels of health associations?
Yeah, and so the answer for that would be definitely. And so Native American health today, they're at extremely high rates compared to the general population for all sorts of things, from their cancer mortality to alcoholism to tuberculosis. We still see a lot of disparities there, and things like underfunding of the IHS, but also the structural issues kind of like we think about in this. If there had been more sort of Native American involvement, perhaps by such a prominent organization, you could imagine a scenario where maybe the facilities might have been more geared towards better Native American healthcare, or a little more tribal input by the Native Americans in terms of maybe stuff that could have helped them in the future. And so, yeah, definitely today there's still a lot of issues for Native American healthcare.
Okay, here's another one. How does Native American healthcare of the past compared to the healthcare of other marginalized minority groups within the United States?
Yeah. So that's an interesting question because, Native Americans really have a separate sort of national healthcare system for themselves, which I think is actually pretty different than what you might see with other marginalized groups, given that they're, they're funded separately through the IHS rather than when you see like a lot of marginalized groups more get their insurance through, for example, like Medicaid or Medicare. And so there's definitely differences, but I think we also see a lot of similarities just in terms of some of the issues that that we find in healthcare of all sorts of marginalized communities, whether it's like not getting the right amount of primary care or like, I think one of the conversations is around like culturally competent care. And so I think that's something that translates throughout different groups.
Okay, we'll just ask one more question then.
Okay.
What surprised you the most, or what did you find most rewarding during the process of doing research?
Yeah, so I thought it was, I was, surprised just how much kind of fun we really had when we got together and we were able to, it didn't maybe not sound super exciting, just like read through like super old documents, but we kind of just like were able to get together as a group. It turned out to be a good experience for all of us, and I really wasn't expecting to be able to like publish or to go to Yale or Montreal, and so those were just a couple of really great opportunities that came to me out of that I wasn't necessarily expecting.
biology major here at Augustana, and this past summer I worked with Dr. Vitiello here in the biology department on the project The Genetic Origin of a Rare Mitochondrial Disorder. And so I'm a biology major, but I'm pre-med, so I knew coming out of my freshman year that I really wanted to get into research, kind of to supplement that journey. And so my 3 reasons for going into research were kind of summed up in 3 words. So the first was application. So I wanted to take what I had learned from my past freshman year and apply it in a laboratory setting. I really didn't have any lab skills going into the summer, but I really wanted to take my science major and actually apply it because I knew that would escalate my learning quite a bit. Connection. So although I want to be a physician, every treatment that you prescribe to a patient likely came from some lab bench or some sort of research or any diagnostic test. So there is a really defined bridge at where research and medicine interact. So I wanted to gain a better understanding of that connection. And then finally is service. So the goal of the job of a physician is to serve the lives of others. And so I really wanted to do that by serving as an undergraduate researcher and also ultimately contributing to the field of scientific knowledge. So kind of a little disclaimer, I'm going to be talking about some pretty heavy molecular biology and cellular biology, so if you get lost, it's okay. I didn't really quite understand everything the first time I talked with Dr. Vitiello about our project, but I'm going to give you a little biology lesson to kind of help you out. So you remember from high school, the central dogma. So you all have DNA. And so what DNA is its in your cells, and your whole entire genome is very vast, is comprised of genes. And so you have 2 copies of every gene. You get a copy from mom and a copy from dad. And so, but it's also interesting to note that your genome is not all coding products. So only 2% of your genome actually makes up you as a person. And how this works is DNA is turned into RNA. And what RNA is it's just another form of information for your DNA template. And then that RNA will get taken by your cells and turned into a protein. And these proteins are what function inside of you and really allow you to function as an organism. So there was a case study of an 8-month-old pediatric patient presented at Stanford Health, and he was displaying symptoms of high blood lactate. And so high blood lactate, what you can think of it this way. So when you work out and your muscles get really sore, that's because your mitochondria, that's the reason you need oxygen, are running out of oxygen. So they produce this weight product called lactate. We'll get a little bit more into the mitochondria part of it. He also was displaying psychomotor delay, cerebellar atrophy, and hypotonia, which is decreased muscle tone. So what his medical providers ordered for him was this diagnostic test called whole exome sequencing. And what whole exome sequencing is so remember I said that only 2% of your genome is actually coded for to make you you. So that is where you sequence just the exomes of your genome, the part that is turning into a product. And it really helps you isolate variables so you can see if there's any variation within your the exomes of your genome. And so what these results revealed was the patient had 2 novel variants in his TARS2 gene. And when I say novel, it means that they have never been documented before in the literature. And it's really interesting to note that this TARS2 gene, the only other documented cases of some sort of TARS2 variant were in 2 siblings who had 2 different variants than this patient, and they both passed away at a very young age, but this patient was still living. So he was a very unique case. So a little bit more about the TARS2 gene. You all have a TARS2 gene, and what it codes for is the protein that is named the mitochondrial preanyl tRNA synthetase. And what this little protein does is it's in your cells, but it will enter into your mitochondria and assist with mitochondria function. So if you remember from high school biology, this is the mitochondria, and it's the powerhouse of the cell. And so it uses oxygen to help you make energy and really just to sustain life. And we know that this protein, the mitochondrial preanyl tRNA synthetase, is really essential for mitochondria function. So because this patient had these 2 variants in his TARS2 gene, we were kind of anticipating that there might be something going on with his mitochondria. So just a little bit more about his 2 variants. One is a missense variant. And so here we have, this is a ribbon diagram of what that protein would look like from the TARS2 gene. It's been created by X-ray crystallography. And so this variant, the missense variant, is specifically impacting the protein itself. So where there should be an arginine amino acid at the 684 sequence, there is a glutamine. And this is in the anti-codon binding region of the protein. So we know, we're predicting that this is going to inhibit partial or inhibit the protein function, causing partial or full loss of function. The splice site variant is the other variant that the patient harbors, and it's a little bit more complicated. So remember, I said that DNA gets turned into RNA. Well, before RNA is turned into proteins, there's these little protein subunits in your cells called spliceosomes, and they will modify the RNA and cut out these sequences called introns that you don't need to code for a protein. And then the exon portion will be left over to code for the protein. Now this patient harbors where there should be a thymine nucleotide, there is a cytosine. And so we are predicting that the spliceosome is not able to properly bind to the intron and pull it out before it's turned into a protein product. So really we didn't know what kind of protein product was being created. So as you can see, he has 2 variants that do 2 very different things. And so that was kind of the nature of his variants that we were going to look into. So here we have a pedigree of the patient's just inheritance pattern. So the proband, that's another word for the patient. So here he harbors his 2 alleles for TARS2. This is his missense variant, and this is his splice site variant. The patient received his missense variant. He inherited it from his father. And so his father harbors this missense allele, but his father also harbors wild type. And so what wild type means is it's what the majority of the population has in their genome. So it's the healthy allele for TARS2. So he had a healthy allele for TARS2, the wild type allele. So he was not displaying symptoms. Similarly, the mother, the patient inherited his splice site variant from the mother, and she also has a wild type allele. So she was displaying healthy TARS2 function. But they both gave the patient a different variant in TARS2. So based off of all this background information, kind of coming up to this summer, our big question that we were attempting to answer was, are these TARS2 variants the genetic origin of the patient's disease? So one of the things that I worked with this summer, I spent a lot of time in the cell culture room here at Augie, and what I worked with are lymphocytes, and what lymphocytes are is they're white blood cells that you all have in your immune system that are really important for keeping you healthy. And so lymphocytes were taken from blood samples from the patient, his mom, dad, and an age match, sex match control. And so we were able to run a variety of assays on the lymphocytes to test molecular biology of the variants and also the cell function. So we really wanted to look at the mitochondria to see if they were being impacted. So here we have some confocal fluorescent microscopy images that we did out at Stanford Research. And specifically, we were trying to look at mitochondrial morphology, so kind of see what they were looking like just from a visual standpoint. Another thing you can do with the cells is you can extract DNA or RNA from them, and then you can send them into companies for sequencing, and they will send you back the transcript of the DNA sequence. And so here we have, it's called Sanger sequencing, and so each little different colored just bump, I guess, looks like it's a different nucleotide. So you can look at what the sequence of the transcript is. Another assay we ran at Stanford was we used this machine called the Seahorse Flux analyzer. And so this allows you to test mitochondria function. So what you do is you put your cells, it's like a 24-well plate. You have 24 wells. You put your cells right here in the bottom, and then a probe is inserted, and it allows you to test a wide variety of mitochondrial functions. So you can test aerobic respiration, which is just basically oxygen consumption, and then you can also test glycolysis and fatty acid oxidation, which are precursors to aerobic respiration. So based off of all these assays, a lesson that I learned from research, and I think a lot of other people learn this lesson as well, is with every result you get, you have to evaluate your next step. And what I mean is, when you go into research, you're testing a hypothesis, and the goal of it is to go into it without bias and to evaluate your results based on what you get. But if you get a result you're not expecting, you really have to keep an open mind and see where you're going to go with your next step. Do you need to reevaluate your process? Do you need to think about your question a different way? So in our lab, when we were testing mitochondria function, we really didn't see a significant difference between the patient and the controls. And then so we tried to count his mitochondria through an assay called qPCR, and we didn't see a difference in the overall number of mitochondria either. So then we had to look at our experimental model, and so we were using lymphocytes, which if you know about lymphocytes, they are an immune cell, but they're not a very highly metabolic cell line. So if you're going to be testing mitochondria function, they might not be the best model for it. So we had to reevaluate, do we need to use a different cellular model or genetically engineer a different cellular model to better look at the patient's mitochondria? So kind of going off of that, we started to assess not so much the mitochondria function, but a different question. So we started to look towards the splice site variant, which remember, we didn't really know what kind of product was being made based on that splice site variant. So the question we turned to was, does the splice site mutation alter splicing? So if you go back to the central dogma, DNA to RNA to protein, remember, the splice site variant, there was RNA, but we didn't know if the spliceosome was properly pulling out the introns to create a proper protein product. So to assess this, we tried to detect this RNA transcript. So what we did was we took RNA from the cells, we reverse transcribed it into cDNA, and then we sent it in for sequencing. And so these were the results we got. So this is the same Sanger sequencing data that I had on that other slide about the lymphocytes. And so down here is the control. And so control harbors wild type. So this is what the majority of the population would look like in their sequencing results. So if you look here at this little G, there's a black peak right here, and that is representative of wild type. The mom harbors wild type as well, so right here. But what we were expecting at this time was that we would not see the splice site variant, so we thought, okay, this is just mom's wild type allele, and then her splice site variant is not showing up. The father harbors the missense variant, which is this little green peak in the background of this black wild type peak right here. But if you look at the patient, this kind of threw us for a loop at first. So the patient harbors a missense allele, which is green right here. But it also, I'm sorry, you guys are going here. It looks like it's a wild type variant. So at first we thought, well, what's going on here? Because we know the patient harbors two novel alleles, not wild type. So we were concerned that maybe our cells were cross-contaminated. But what it turns out, we kind of had to just think about it a little bit more, and what we were looking at was actually what we had been looking for the whole time. So another way you can visualize DNA or RNA is through gel electrophoresis. So what gel electrophoresis is you take your RNA, you reverse transcribe it into cDNA, and then you make a lot of copies of this DNA transcript through PCR amplification. And then you run it on a gel, and it separates out your DNA by size. So the smaller DNA will end up down here, and larger DNA products will end up here. And so if you look at these wells, this is the mother, father, and the patient. This is the mother's wild type allele. So if we were to look at an mRNA product that contained the exons 2, 3, 4, and 5, it would be approximately this size. So this is correctly spliced. Father, this is his wild type allele, but it's also probably his missense allele, because remember that was only impacting the protein function. And then here in the patient is his missense allele as well. But if you look down here at, this would be about 190 base pairs in size. There's a band in the mother's well and also in the patient's. And so upon further analysis, we found that if you were to splice out exons 3 and 4, you would get a band that is approximately 190 base pairs in length. And so that is what you are seeing right here, which actually turned out to be the splice site variant. So what kind of is concluding on these results? This is just some more Sanger sequencing, and this is the splice site variant, this black peak right here, that we were able to detect. And so the gel electrophoresis revealed a band that was 190 base pairs, which if you were to pull out exons 3 and 4, you would get a product of this size. And so what was happening, what we had thought is that maybe before in the research, that the intron was being retained, but actually what was happening is the spliceosome was cutting out parts that you really need to make a proper protein. So the next step in our research would be to analyze this mRNA sequence and to maybe produce a theoretical protein product and then analyze the protein. So I started out with 3 reasons for going into research, and then I'm going to end with 3 lessons that I learned from research. So probably one of the biggest ones was persistence. So the challenges you work through now may lead you to the answers. So when you're in the lab and you are running an experiment and you get a result you weren't expecting, sometimes that can be really, really frustrating. But you have to be able to step back and keep an open mind because, well, maybe you need to rerun your experiment, but also maybe you need to think about your problem a different way. So like us, we had to look at that splice site variant and evaluate the data we had, and then think about it a different way, and we found, we're able to come to a conclusion. Or with the lymphocytes, thinking about a different cellular model. Learn as you go. I mean, I came into this as a freshman. I really had no lab experience at all. And I was surprised within that 10-week period that I did this research, how much I was able to learn and how many skills that I gained. You do make mistakes, but by the end of the summer, you will be really amazed at how much you know your project and you can apply it in a classroom setting as well with the knowledge you gain. And then finally, service. So I started out wanting to go into research for service, but I ended. That was probably one of the biggest takeaways I got from this. Because you got to remember the reason that you're doing it. So when you're in the lab running an experiment, it's really easy to not really remember why you're doing what you're doing because it seems so separated from the rest of the world. But in reality, we were trying to give this patient and his parents an answer about this little boy's variants. And also, really, you contribute to the field of scientific knowledge so other doctors and researchers can pull from that and add to it. And then potentially you revise treatments and overall are able to improve the health of others. So, acknowledgements. I'd like to thank Dr. Vitiello, Matt Mose, who I worked with this summer, the past lab members, Brooklyn Breeze, Luke Bartle, and Josh Ryder, our collaborators at Stanford Research, and the Augustana University Biology Department, Spur, Grin, and Epsilon for the funding.
Okay, I'm going to combine a couple questions here for you. So one was, where does the patient go from here? Is there a treatment for them? And the other question combined is, if there isn't a treatment, then how is your work important for this?
So our contact with the patient was extremely limited. So we worked with the cells, so we had to go through an IRB to go through that training because it is a human cell line. But as far as the patient, I don't really know that much about him, so I can't really speak for the patient. But how this work is important and pertaining to the patient. One, it's important that we learn about these variants so we can provide information to the patient, his medical care providers, and his parents. But also, like I said, ultimately you're contributing to the field of scientific knowledge. So if another case of TARS2 variants is ever documented, similar to this, it's probably going to be used as a helpful diagnostic tool to help that patient get the care they need faster and ultimately improve the quality of their life.
Okay, you might have to explain what this question means. Did you utilize HeLa cells in your research?
HeLa cells? Oh, Henrietta Lacks cells? No, we did not. The cells that we worked with were taken from blood samples from the patient, parents, and a control. So they were just lymphocytes.
What are the current limitations of DNA editing to cure genetic disparities?
I mean, I don't really, DNA editing, wow, okay. So you're going from, DNA editing can go anywhere from like GMO products all the way to human healthcare applications, which as far as I know is not really in practice right now because there are, if you're talking about like CRISPR, that kind of genome editing, there are a lot of, what's the word I would use? It doesn't work super well yet to apply in a clinical setting. So I would say that it's not being utilized yet. I can't really speak very in-depth on that topic, but as far as like our lab, if we were to use some sort of like genetic like modification thing, it would be to take like stem cells from like body stem cells, and then you can turn them into like like induce the mutations in that and then study from that, but we haven't done that, so.
Okay, I have one final question. You mentioned there was another case of siblings who died early in life. Can you say why this patient is still alive and the others died?
So, like I said, those two siblings had two different variants in their TARS2 genome. So it's the same gene, but a totally different set of circumstances. So I don't really know the specifics of their case, but it could be with this patient. Again, we're still trying to connect those variants to what the patient is experiencing as symptoms. It could be, the environmental impact of how the patient is living his life, or the patient might have partial function of his TARS2 gene producing a partially functioning product, so he's able to live off of that, so adapt that way.
Well, hello everybody. It's nice to see you all here. My name is Nolan Ortbahn, and I'm a freshman here at Augie. I study science education, and this is my presentation, India, Engulfed and Excrement. Over 200,000 children in India die from fecal contamination diseases every year. Let that sink in. One of the most rapidly developing countries in the world cannot care for the basic needs of its people. But why? The answer to that question could save hundreds of thousands of lives every year. So I went searching. My research began in Dr. Daniel Gerling's FYS 110 class, Fecal Matters, and the need for a topic for my first paper. In Dr. Gerling's FYS class, we studied things like the American sewer system and the global sanitation crisis as it relates to third world countries. I knew I was going to India in January with the Dharma trip led by Dr. Looney and Dr. John Pennington, where we studied life, religion, and culture in contemporary India. So it made sense to study something else that would have some practical application. I'm also very interested in the science field, as I want to be a high school science teacher, and most specifically I'm interested in public health and epidemiology, so it makes sense to study sanitation because of how closely it is intertwined with public health and epidemiology. But once my research began, I was quickly made aware of the severity of the situation. Millions of people struggle with the consequences of open defecation and unsafe sanitation systems. So what is the government doing to stop the problem? Surely the Indian government must have some role in trying to curb this issue and reduce the number of deaths every year. And they do. They have a recently administered program known as Swachh Bharat, or Clean India. The goal of this program is to have the country free of open defecation by the year 2019. A lofty goal, to say the least. The program was started in 2014 by Prime Minister Narendra Modi, with the goal of building toilets all over the country while educating Indians about the dangers of open defecation. And this process is pretty good, but the use of shame to ensure the use of latrines is controversial. I adamantly supported shaming as a technique in my preliminary research. So I'm going to describe the process of shame and how closely it works with the sanitation problem in small villages. So oftentimes, the government will go into a small village with the means to build these latrines. And they'll go and they'll do a session with the villagers, and they'll say, okay, here's why you need a latrine, it'll help protect your citizens, it's the morally right thing to do, we think it makes sense, so we're going to build these here. And the villagers get together, and more often than not, they go and talk to the government employees and say, look, we've been doing this for thousands of years, Buddha didn't need a toilet, why do I? And so the government goes to leave, but before they do, they say, okay, we understand where you're coming from, but you're awful, disgusting human beings for wanting this, you're killing your own people, how could you? This is ridiculous. And so the people feel ashamed of what they are and what they've been doing. And so they'll go to the government, wait, we understand now the severity of the situation. We understand now that we need to change. We want these latrines. And I supported it. I thought, a life saved is better than a life lost, no matter the circumstances. I thought Swash Bharat was going to be India's saving grace, before my field research in India revealed a different side of the story. So of course, when I went to India and we landed in Delhi, I was super curious to see all of the sanitation systems in the airport, because that was going to be my first mode of contact. And so we got off the plane and I went straight to a bathroom. And this is what I saw. As you can see, it's pretty basic, pretty much a hole in the ground, but there is a flushing mechanism on the wall that you can't see. But it's very clean, it's very well kept and very well maintained. I was surprised. I had done all this research about how awful Indian sanitation was, and so I was very shocked to see how nice the facilities were in the Delhi airport. Much of the same is to be said about the toilet in our first hotel room. A little bit more standard looking, with like a seat and a lid, flushing mechanism there up top, but still very well maintained, very clean, and I was beginning to be skeptical. Was the Indian sanitation crisis really as bad as it seemed? And of course it was. And I realized this in a village just outside of the city of Haridwar. And this is where I realized the severity of the sanitation problem. So as we began our tour of the city, you see these huge amounts of standing water right next to the road. And this water often comes from major rain events. There's no curb and gutter system to drain the water. There's no sewer system to deal with it. So it just sits there, and it can sit for weeks on end. And when it sits there, dangerous bacteria can begin to fester, and with contact with the skin or open wounds, it can be potentially life-threatening. As you can see here, right near the same body of water, we have a couple of kids wearing open-toed shoes. And of course you can see that to walk across the street, they would probably have to walk right through that water. And if they have open wounds or any exposed flesh, it could prove deadly, the bacteria that lies in this water. Next on our trip, we got to visit the house of a woman. And Indians are very proud of what they have. She was super excited to boast about what her family had built and their time in this village and all that they were able to acquire. And she even boasted a bathroom. And this is their bathroom. It's pretty rudimentary. It's literally just this basic. And so what would happen is, as the day starts, family members would use this basin, relieve themselves, and then when it fills up, somebody would walk it out to a field, dump it out, and bring it back. And this sort of toilet is the norm in India, especially rural India. This is actually probably an above average amenity, as most Indians would walk out to the field each time they had to use the restroom. So this is fairly average. This isn't that shocking, and this is what I expected to see here in this village. And the excrement from some of these pans ends up in the standing water that we saw alongside of the roads. Because this family was courteous enough to walk it outside of the village. Some families would just toss it out the door. And so when you have a rain event, you run into those ditches, bacteria is bound to spread. I now fully realized how dire the situation was in India. So later in the day, one of the last things we did in this village was visit this school. And on the outside of the school, they had this chalkboard. And written on the chalkboard was Clean India, Healthy India, which is kind of the mantra of Swash Bharat. And India is trying to educate its citizens about sanitation as early as grade school, teaching students the dangers of open defecation to persuade kids to practice safe sanitary procedures. India's government understood that you can't start with adults. That's not going to institute any change. You have to start with the youth, and that's where the movement began. And so I noticed this inscription on the chalkboard, and so I asked a girl from around the area about Swash Bharat and her impressions of it. And she raved about the program. She thought that Prime Minister Modi was doing a fantastic job, that Swash Bharat was certainly on its way to achieving the goal of open defecation-free India by the year 2019. This blind sort of belief that I had actually before I had gone to India and seen the problems firsthand. And her dialogue got me curious to see if her belief echoed those of the rest of the country. So I talked to our guide, Sushil Bansal, about his take on the matter. It's important to note that Sushil is a staunch supporter of Prime Minister Narendra Modi. Sushil is a Hindu nationalist and supports most of Modi's legislation. So I assumed that Sushil was going to also rave about this program, but Sushil saw the flaws. The building of latrines, by his estimation, is not going to do enough to reach the goal of 2019, of Clean India by 2019. Sushil knew that the government was doing a lot, but it was never going to be enough. This problem is so monumentous that just building a few latrines here and there and trying to tell some people to change is never going to result in anything positive. Sushil did talk about, though, the effectiveness of TV ads. So here's one of the TV ads or a sign you might see on a billboard throughout India. One step ahead to a clean India. And of course you have India's hero, Mahatma Gandhi, draped in the Indian flag. They're trying to instill this sense of patriotism and pride. This is India's fight. This isn't each individual's fight. This is India's fight. And so after the interview with Sushil, it became clear to me that swashbuckling is not at all what it's made out to be. The program has utterly failed its goal. The program will absolutely, unequivocally not result in Clean India by 2019. The building of latrines just isn't enough. The education of these students and these citizens is not going to be enough to get wholesale change in just 5 short years. But they are using different media as a form of spreading the message, which could be the start of something great. Toilet the Movie was a huge success in India in 2017, actually their best-selling movie. You could compare this to the latest Marvel or DC films in the United States. It was wildly popular. It's a love story centered around a man's love interest, and she won't move. She refuses to move into his house until he has built a toilet for her. And this makes sanitation a more relatable issue for everyday Indians. But it's also created a moral divide between urban and rural Indians. So this movie, the people that could afford to see it are oftentimes the wealthier Indians. They live in the bigger cities, the more urban settings. And so they're getting a lot of this, oh, you know, the sanitation problem's really bad in rural Indians. How can't you see these problems? Like, we have it figured out here in the cities pretty well, but it's still a problem in rural India? Come on, you guys can't figure it out? This is kind of ridiculous. And so think about it like we live in South Dakota. So we live in a very small, rural state. If we were doing similar things, costing the lives of our people, and urban folk from New York or LA were saying, oh, how could you? You're so stupid. Don't you understand what you're doing? Like, are you not globalized enough to realize the debt that your actions have? And we wouldn't listen to them. We would turn our backs and continue doing what we've been doing for hundreds of years. Urban Indians seem to be harsh on villagers for not understanding the consequences of their actions, causing enough shame already. India needs love, not cruelty. And I don't have an answer to the sanitation crisis in India, but one thing is clear to me. India is a beautiful country with beautiful people. I feel a connection to the Indian people unlike anything I could have ever imagined. We stepped foot in their country, and they welcomed us with open hearts, open minds, and open arms. Into a culture which we were unfamiliar with. We didn't know their language, we didn't know their religion, we didn't know their day-to-day lives, but that didn't matter. They welcomed us with open arms, and for that, I will ever owe a debt to the Indian people. The experience has totally changed my outlook on life in other countries, and has helped me realize how blind Americans are to problems around the world, proving how important field research is in forming an opinion. Without hands-on experience, research is just words. My preliminary research in FYS 110 initiated my passion for sanitation, but the research I did in the field and on-site was invaluable to my knowledge and understanding of the topic. My personal hands-on experience even caused me to amend my original opinion on shame and its effectiveness in India. Without the personal experience, I would never have realized how important compassion is to changing the status quo in India. And of course, none of this is possible without the liberal arts education I received here at Augustana. The liberal arts education provided by Augustana emancipated my free thinking, uncovering the social truths of the world with which the humanities ponder. I thought for sure my time at Augustana was going to mostly be spent in an education classroom and in a science lab. But when I found this passion in an FYS class, it opened my mind to critical thinking and thinking about the social problems in the world as the humanities does. Study abroad was also key in my experience, and it's morphed me into a more conscious humanitarian. Learning the intricacies of life in a different country awakened me to problems that go deeper than the surface of the country. Without my experience abroad, my ignorance to Indian culture would never have been resolved. Certainly, studying abroad, learning about a people that are different from you is invaluable, whether or not you are doing research. It worked out well that this awesome trip to India that Dr. Lenny and Dr. Pennington led coincided with my research, and so I was able to capitalize on two events at one time. And finally, research. I never thought I would do any research here at Augustana until I was an upperclassman, but I was just flipping through pages of articles and pictures of India when I took interest. Dr. Gerling helped me to connect these resources I needed to turn this FYS paper into research abroad. This research has stimulated my critical thinking and pushed me to a topic which touches me deeply. India is neck deep in the messy process of reform. Change takes the effort of each and every citizen digging toward a happy and healthy country. With open hearts and open minds, India can scoop its way out of this mess once and for all. Thank you.
Okay, here's your first question.
All right.
Does the sanitation crisis in India affect men and women differently?
It does. Women are disproportionately more affected by the sanitation crisis in India. Not necessarily scientifically, but their risks of using the restroom out in the field are much greater. When women have to go into the field and use the restrooms, it is customary that they cover their face. And so this can create problems. You can get snake bitten, which happens more often than you might think. Women are raped very often when they use the restroom out in the field. And so the process is just a lot harder. So men also have to deal with the health risks, but women also have these social risks that go along with using the restroom out in the open, which is key to making sure the latrines are private and you have the ability to be by yourself, use the restroom in a safe and secure location.
If Clean India isn't working, then what other things could India do to help with this sanitation crisis?
I think Clean India is working to an extent. It's not going to reach the goal of open defecation free by 2019, certainly, but it is doing good things. I don't know that I have any answers. I didn't spend enough time, researching. I was only in the country for 26 days. I learned what I could, but certainly this problem is bigger than me, and is as big as India itself. And I think that by my estimation, if India is going to have any sort of cultural change, it's going to have to be through media. Toilet the Movie is going to start this revolution, and I think that if they can keep pursuing the use of multimedia in different ways to affect change, they can have this wholesale change as a country within the next 50 to 100 years.
What is something that we can do here to make a difference?
So my advice to each and every one of you would be to help lift the taboo of going to the restroom here in the United States. The taboo is very similar in India, but we don't like to talk about it much. We don't talk about going to the bathroom. We don't talk about GI tract problems or problems with urinary tract infection. They're touchy subjects. We don't like to talk about those. And as long as the taboo stays, we can't reach this enlightenment that we need and that India needs to be able to solve these issues worldwide. So feel free to talk about going to the restroom. Talk about these issues, because that's how we're going to get change in the United States and certainly in India.
I think that's all the questions we have for you. Thank you.